Ronald Worton was born and raised in Winnipeg.
My PhD supervisor was James Till, and he was wonderful to work with. There was an added plus that he worked closely with Bun McCullough, who also was a terrific supervisor, and in my case, a sort of second supervisor. After I left the stem cell research as a PhD student, I went over to Yale to work on more biochemistry, to learn some new technology. And I came back to the genetics department at Sick Kids, hired by Lusa Minovich, who's another laureate, and spent my first 4 or 5 years establishing a diagnostic service lab.
When he moved to Toronto, he totally revamped the cytogenetics lab here and made the cytogenetics lab at Sick Kids one of the foremost cytogenetic labs in the country, with a significant reputation outside the country.
The issue with muscular dystrophy was that there was a ton of research being done and nobody had any idea what caused it. It was a neuromuscular disorder. It wasn't even known whether it was caused by a problem of the nerves or a problem of the muscle.
And the first evidence that he needed to discover the cause of muscular dystrophy was a female who had muscular dystrophy.
And she had a translocation, which is a rearrangement of the chromosomes between her X chromosome, which we knew carried the gene, and another chromosome.
What Ron did was particularly innovative because he found a girl who suffered from muscular dystrophy, which is unusual, and he was able to identify the fact that her chromosome was broken, so he could take this bit, and because he knew what this bit was, he could identify the gene by walking essentially from this bit to this bit. And he had to design some special experiments to be able to do that.
Cloning of the gene enabled us to be able to do prenatal diagnostics with much more accuracy than had been possible before.
The diagnostic tools using the gene would have originated in Toronto in my lab and in Boston in Lou Kunkel's lab at about the same time. And then we sent our probes around the world and everybody adopted those tools.
The impact that this research has had is enormous around the world, because it means that since this disease occurs in every single population, that we can now do a diagnosis and carrier detection for all families wherever they are.
Our lab in Toronto did the world's first prenatal diagnosis in a woman who had a nephew with the disease and had already terminated 4 pregnancies of males because she was afraid that they would have the disease. And we were able to demonstrate that this pregnancy was a male who was not affected, and she went on and her baby was perfectly normal.
And so not only did it lead to better understanding of the disease, but it also revolutionized technically what you could do in human genetics to identify new disease genes.
Well, there's 2 things I'm most proud of. One is the discovery of the gene for muscular dystrophy because of the tremendous importance it had in everything else that followed. But the second one is the creation of the Research Institute at the Ottawa Hospital that now is one of the foremost medical research institutes in the country.
He used his position to build additional networks across Canada, like the Genetic Disease Network, Stem Cell Network.
It's Ron's generosity and general collegiate spirit that enabled the field to move very quickly after he'd made the discovery, because he was always sharing his data with other people.
An outstanding basic scientist, visionary leader, and genetic trailblazer. Ladies and gentlemen, Canadian Medical Hall of Fame laureate, Dr. Ronald Worton.